Girls, can you see this screen, ma? Hello?
Yes,
Girls, can
ma'am.
you see the screen? Yeah.
Yes, ma'am.
Yeah. What is this? What is this plant name I have written over there?
Marselia.
Marselia. Okay. Now we'll begin with the class. Right. So this is the Marselia plant, which is belonging to the Pteridophyta and Pteropsida, the order Pteropsida, it belongs to the class Pteropsida and the order Marceliales and the family Marceliaceae. Okay, that is a classification about the Marselia. So it is commonly called as this Marselia plant. It is commonly called as water ferns. This is commonly called as water ferns. Now, what do you mean by here water fern? That is a pteridophytic plant. So this is an aquatic pteridophytic plant. Aquatic ante they grow in the water. Okay. So the pteridophytic plant, an example of pteridophyte, which is an aquatic ante, it is a Marselia. So as it is growing in water, so the name called it as water ferns, commonly called as water ferns. Now, it is also common in the temperate regions and also in the freshwater ponds and ditches. And the best example what we can find is Marselia quadrifolia. So you can see here the species name quadrifolia. Folia is always refers to the leaf. Quadri means four. You can see this plant. It is going to have the 4 leaflets. So you can see here. Okay. So based on that structure, the structural of the leaf, the species name is called as the quadrifolia, Marselia quadrifolia. Now, so we'll go with the general structure, that is a morphology, external morphology of the plant. Now the plant is a sporophytic plant. It is a sporophyte. That means they are going to develop sporangia. Now, the plant body, it is differentiated into roots, rhizome, and the leaves. There is no stem as it is a free floating. It can float on the surface of the water. So we can see only the presence of the rhizome. And underneath the rhizome, we can see the development of the roots and the leaves. Now, when you talk about the rhizome, so here this is an aquatic plant where the stem is get reduced. Okay, we can't see any stem over here. The stem is completely getting reduced in the form of a rhizome. Next. Now, this rhizome, it is dichotomously branched and it is going to have nodes and internodes also, and a number of adventitious roots which will be arising at each node on the ventral side, whereas at each node we can see the development of a leaf which is coming from that each node. So let me show you the picture over here. So you can see here, this is the rhizome part. Okay. So here, this is the one which is growing horizontally. This is the rhizome. Now each rhizome, here I'm showing you, this is the presence of the nodes, where from each node on the ventral side, that is below the node, we can see the roots development. A cluster or the fibrous roots will develop. And on the upper side, that is on the dorsal side, we can see the formation of the leaf. This is a leaf where the leaf is getting dissected into this 4 leaflets. Right. So that is a characteristic feature of the Marselia. We can identify the Marselia plant with the help of these 4 leaflets. Now, how are the leaves? Leaves are compound. Why I'm saying leaf as compound? Because it is dissected into 4 leaflets kabati. Now, each leaf, it is going to have a long petiole and 4 carrots. That means 4 leaflets, we can call it as. The carrots, they are arranged in cross-like manner at the tip of the petiole, and each leaflet is triangular in shape. The veins, they are going to form the reticulate arrangement, and the stomata are located always on the dorsal side and also on the ventral side of the leaflets. So you can see the picture over here. Right. So this is a long petiole of the Marselia, which is arising from the node of the dorsal side of the rhizome. Now, the tip of the petiole, we can see the leaf, which is dissected into these four callets. We call them as callets, or we are also calling them as the leaflets. Now, how they are arranged, you can see here. They are arranged in the form of a cross. Two leaflets, they are opposite to one another. Okay, they are the cross, in the form of a crossly they are arranged. And then each leaflet, you can see what is the shape of the leaflet here. Each leaflet is a triangular in shape. That is a characteristic feature of Marselia. Triangular in shape, and each leaflet we can see the formation of the midrib region, or that is a reticulate venation. Right? So that is a and on the dorsal side as well as on the ventral side, we can see the development of the stomata, so which are useful for the exchange of the gases. Now, next we'll go for the roots. So when we talk about the roots, how are the roots, Amma ikkada? How are the roots etla unnai? Are they taproot or are they fibrous?
Fibrous.
Fibrous, that is, they are the adventitious root system. Now, after that, so we can see here during the reproduction, from the base of the petiole, you can see some bean-shaped structures which are arising over here. These are called as the sporocarp. Now, these are the one which are organized from the base of the petiole. They arise from the base of the petiole. Okay. Now, so these sporocarps are the structures which are going to produce the spores at the time of reproduction. Okay, so they'll be having a stalk. So with the help of the stalk, they are attached to the base of the petiole. So generally, each that is generally in this species, in general species, we can see 2 to 5 sporocarp. That is in Marselia quadrifolia. When I talk about, we can see the development of 2 to 3 or 4 to 5 sporocarps developing at the base of the each petiole. Now, so another important point I want to tell you here is the younger leaves. You can see here the leaves which are at the younger stage of the leaf, they are undergoing coiling like this. Okay, so can you tell me what is this feature called as the coiling of the younger leaves? In the lycopod, that is a characteristic feature of the pteridophytes. In the general characters also, I told you that. The coiling of the younger leaves, it is called as circinate vernation. Okay, so that can be clearly observed here in this Marselia group. Now, roots are of fibrous type, which help in floating the plant on the water surface. Now, we'll go for the internal structure, that is the anatomy. So, first we'll go with the anatomy of the rhizome, which is very, very important in this particular structure or in this particular plant. Now, the rhizome, when we talk about the rhizome transverse section, we can see the following tissues, that is the epidermal tissue, cortex, and the central stele. Now, so epidermis, it is the outermost layer, or it is going to form the outer covering, whereas the cortex is wide. That means more portion, majority of the portion of it, of this rhizome is occupied by the cortex. And its a peripheral part, that is the outer part of the cortex, is made up of parenchymatous cells. And in this parenchymatous cells, we can see the presence of a large number of air chambers. This is referred to as the parenchyma, so which is a characteristic feature of the hydrophytes, which stores the air. Okay, these are called as parenchyma, the parenchymatous cells, or the tissue which are having these air chambers, which are storing the air, that we call it as the parenchymatous tissue. Now, the inner portion of the cortex, it is composed of sclerenchymatous cells, and the endodermis is going to be present inner side of the cortex. So you can see this, the picture, the transverse section of the rhizome, Marselia rhizome. So you can see this is a single layer of epidermis which forms the outermost layer. Below that we can see this is the parenchymatous cortex region. This forms the parenchymatous cortex region in which we can see the presence of these air chambers. This is the air chamber, or we can also call them as air cavities. So this tissue is designated as parenchyma. So which is helpful in exchange of the gases or the transportation of the air or storing of the air inside. Next is the inner cortex. Here you can see this is made up of the sclerenchymatous tissue. You can see here the walls are been highly thickened. So this forms the inner cortex and producing this the sclerenchymatous tissue. Now below that you can see the formation of the endodermis layer, and internally you can see the stellar steel. Okay, now the steel in Marselia, it is of amphifloic siphonous steel, and that too it has pith in the center, and protoxylem groups are exarch in position. Now what is this amphifloic siphonous steel? So as we say that the central part of the steel here, it is occupied by the pith region. Okay, the central part of this is occupied by the pith region, and the, what do you call, the xylem, you can see here. This is the xylem tissue. Okay, and the xylem getting surrounded by the phloem. Okay, it is getting divided into inner xylem and the outer phloem and all that. Whereas the central part is occupied by the pith region. So it is called as the amphifloic siphonostele. Okay, so and the protoxylem, what we talk about, the protoxylem is of exarch nature, and it is varying from one species to the another species. So this is a very important question in the exam point of view, the internal structure of the Marselia rhizome. Now we'll go with the internal structure of the leaf also here. Now when we talk about the internal structure of the leaf, so both the surfaces of the leaf, they are bound by the epidermis, upper epidermis and the lower epidermis. And the epidermis is covered by cuticle. This is again a characteristic feature of the hydrophytes. And it is going to have the sunken stomata in the epidermal region. Basically, they are present in the lower epidermis. And the mesophyll cells in the center are of between both the epidermis. That mesophyll cells are differentiated into palisade and spongy, just like the higher angiospermic plant. So these forms the palisade tissue, what I am drawing here, and below that you can see the spongy tissue or the spongy cells which are very loosely arranged. And in the center you can see the presence of the vascular cylinder where the xylem is going to be surrounded by the phloem, which is a siphonostelic condition, and it is owing only a single vascular bundle. That's why a single vein passes through the each leaf. So this is the internal structure of the leaf. And one more point is also there I have not mentioned over here regarding the transverse section of the petiole. That is also very, very important. So that in the next coming slides, I'll show you that. So before that, we'll go for the reproduction. So in the Marselia, during the reproduction, they are going to develop, okay, the very important fruiting body-like structure, what we call it as the sporocarp. Okay, so this is concerned with the reproduction. So, but they also reproduce with the help of the vegetative reproduction also, which is carried out with the help of the tubers. So tubers are the one which are helping in the plant to carry out the vegetative reproduction, which are going to store the food material, and they utilize this food material and they can transfer. So these tubers, they will develop into a new plant. Now, when we go for the sporocarp, now what is this sporocarp? So this is a structure which is a fruiting body like structure, or which is a bag like structure, or a sac like structure, which are developing the sporangia. So very, very important. So when we talk regarding the sporangia or the spore development here, Marselia is a heterosporous plant. I repeat this. Whereas the other two genera what we studied, that is Lycopodium and the Equisetum, they both are homosporous plants. But whereas the Marselia, it is a heterosporous plant. That means this plant will develop and produce two different type of spores. The spores which are larger in size, we call them as megaspores, and which are smaller in size, we call them as the microspores. So they will be developing these spores, microspores. They are produced in the sporangia, which are called as megasporangia and the microsporangia. Now, these both types of sporangia, they are found within the same sorus or the sori. Okay. Now, this sori, they are produced in hard fruit bodies, which are called as the sporocarps. Now, what are these sporocarps here? They are having the stalk, which we call it as the peduncle, and they are going to be attached to the base of the petiole. So in the comic picture, చూపించాను కదా. So you can see here. So this is the sporocarp. So this sporocarp, it is going to have, this is the stalk, what we call it as the peduncle, through which it gets attached to this long petiole. Now, so how is the sporocarp shape? It is appearing just like a bean. Bean అంటే మనకి చిక్కుడు గింజలు ఆకారంలో ఉంటాయి అన్నమాట. This గింజ ఆకారంలో. So bean shaped structures, it is a hard, and so when we look at that, the covering of that sporocarp, it has hard and stony wall layer. Okay, it is very hard. It is not as soft as such. So that we call it as the capsule. Now its wall has a single vascular bundle for supplying the food material to the sporangia. Mute yourself, ma'am. You can mute yourself. Please check who is on mute. Just mute yourself. Now the sporocarp, when it is observed that inner side, it has two inner chambers. Now each chamber, it has a row of sori. Sori are nothing but the sac-like sporangia, we call it as. Now the sori of two rows alternate with one another, and it is formed by the wall of each sorus is formed by its own indusium. Indusium is nothing but it is a nutritive layer supplying the food. Now each sorus, it contains a row of megasporangium and several microsporangia. We'll show you the picture over here like this. So here sporocarp structure is the pedicel by which on stalk we called it as, which is getting attached to the petiole, and this is a bean structure. Outer wall, this is the outer wall of this sporocarp, which is very stony and hard, what we call it as the capsule. Now this sporocarp is entirely supplied with only a single vein. So this is a single vein which produces which will help in conducting the water and the food material. Now, so when we take the transfer section, when we take here the transfer section of this complete sporocarp, so you can see the presence of these sori. So these are called as the chambers of this sporocarp. So this is one chamber. This is another chamber. Okay, this is another schema. These are called as the sori or the bag or sac like structure which are developing. In this we can see the formation of the sporangium. Okay, now so this is the sori here which is going to accumulate the sporangia. Now you can see here each sporangia has its own nutritive layer or the covering layer, what we call it as the indusium. Okay, it's ఇక్కడ చూడండి మీరు ఈ సోరై చుట్టూరా ఒక wall layer లాగా ఉంది. So that is called as the indusium. This is a nutritive layer supplying for the sporangium. Okay, so here each sori. Next. So inside the sora, you can see the formation of the larger circle you can see inside. So that one is called as the megasporangium. And at the base of it, you can see the microsporangium. So many microsporangium can be developed and very few are the megasporangium. Okay, the smaller one, what you can see, they are the microsporangium, and the larger one, what you can see, that is the megasporangium. So this is the structure of the transosection of the sporocarp. So you can see here this one also. Okay, sporocarp, the outer wall layer. This is the outer wall layer. This is the epidermal. This is the midrib region which is supplying the conduction of the water is going to happen through this mucilage canals on either side of it. Now this is a mucilage canal here, and this is a mucilage canal, and this is each one. So this is the each one which we call it as the sori. Any sori? 1, 2, 3, 4, 5, 6. On they both, each sori, they are alternate, they are opposite to one another like this. Now inside this sori we can see the development of a larger sporangium, what we call it as the megasporangium, and smaller sporangia which are called as the microsporangia. You can see here this is a micro and this is a megasporangia. Now next, a large placenta is produced on the inner side wall in the young sporocarp. Now the placenta of two sides, they alternate with each other, and the megasporangia are produced on the same place, and each placenta is covered by its own individual endosium. Okay, megasporangia will develop, mature earlier. So when you talk about this, the megasporangia, it will mature earlier when compared to the microsporangia. That means it is going to develop the megaspores earlier when compared to the microsporangia. Now each megasporangium contains only a single megaspore on maturity. But each microsporangium, they contain about 30 to 64 microspores in it. So you can see the difference. Though the megasporangia is large, but it is going to develop only one megaspore. Whereas the microsporangia, it is very, very small, but it produces as many microspores as it can inside the microsporangia. So that is a major difference lying between them. So you can see here, this is a megasporangium. So just now the same one. Now, so development of the sporocarp, how this sporocarp is going to be developed. So sporocarp, it is believed that it is a single folded pinna. That means to say it is a leaf-like structure in the beginning. They imagined. But this pinna, it has a single vascular bundle, and it is going to have the receptacles or the placentas.
Zero dali the painkiller tablet in a baby.
So receptacles or the placentas. Hello. Please mute yourself, ma. So these receptacles and placenta, these are considered as the base for the attachment of this sporangial initial. So from these receptacles, the sporangial initials will develop. Now these sporangial initials, they will develop into sporangia. Okay, next. So the dehiscence of the sporocarp. And the sporocarp, etla break అవుతుంది to release the spores. The mature sporocarp, it open after 2 to 3 years of its development. That means when it makes sure that all the spores are ready inside the sporocarp, then it will going to break. That we call it as dehiscence. So it will open only after 2 to 3 years. Now a stony wall. So I told you that they're going to have a stony wall or a hard covering layer of the sporocarp. So that we call it as a stony layer. So it will undergo decay. Decay ante adi baga endipoyi adi pagultundannamata and open the sporocarp. Now the inner gelatinous material, whatever is there, it will absorb the water and swell. As a result, the sporocarp will split into 2 halves and the spores which remain viable for a very long period. So adi break ayinappudu all the spores which are present inside that, they will just break up and form the spores. Now how this sporangia are developed, you can chudandi ikkada. The sporangial initial, it cuts off a jacket initial to the outside and an archisporial initial towards the inner side. Jacket initial, it produces the single layered wall of the sporangium, and the archisporial initial, it cuts off into 2 tapetal cells. These 2 tapetal cells divide to produce 2 layered tapetum, so which is an issue. Now the archisporial initial then divides and produces 12 to 16 spore mother cells. Now each spore mother cells, they undergo meiosis and produces the spores. Okay, so that means to say within the sporangium, suppose I'm talking about a microsporangium. The microspore mother cell will undergo reduction division and produces 4 microspores, whereas in megasporangium, it will undergo reduction division and produces the megaspore, a single megaspore. Now the tapetal cells, it is going to provide the nourishment to the younger spores, and so they disintegrate. So after the development of the spore, this tapetum will get disintegrated. In the megasporangium, I told you, just only 1 spore is going to be developed, whereas the other remaining, all the cells, they get degenerated. Now the mass which are developed is the nourishment to the developing megaspore, whereas the microsporangia produce around 30 to 64 microspore. Now, next, another important point here is these microspores and the megaspores. By breaking of the sporocarp, they just fall onto the surrounding environment and develop into the next generation. We call it as the gametophyte. Now, what is this gametophyte here? The microspore will develop into male gametophyte and the megaspore will develop into female gametophyte or a female gametophytic plant. Microspore will develop into male gametophytic plant. So how is that? You can just see now here. So this is a microspore, okay, which is developed from, or which is produced from the microsporangia. Now this microspore, first it undergo one unequal division. Unequal and it is going to form one bigger cell and one smaller cell. This smaller cell, we are calling it as the prothalial cell or the first formed cell under. The upper one is called as the apical cell. Now, this apical cell here, it undergoes again one transverse division and forms 2-celled stage, which are considered as the antheridial initials, or we call them as the antheridial initial cells. Now, next afterwards, this antheridial initial cells, well, one of these 2 cells, again, they undergo one periclinal division and produces the jacket layer of cells here, and these are the antheridial initials. Next, they keep on dividing and dividing and producing the second jacket layer of the cells and the third jacket layer of the cells. And at the end, they are going to develop into the antheridium or the anthrocytes. So you can see here. So this microspore, first it divides into 2 cells. First smaller cell, we are calling it as the prothalial cell. Next, the larger cell emanamu. Larger cell is the apical cell, which is dividing into two antheridial initials. Now, each antheridial initial it divides to form three jacket cells and a single androgonial initial. Now, this androgonial initial of each antheridium divides and produces 16 androcytes or anthrozoid mother cells. These anthrozoid mother cells, there are further produces into antherozoids. Okay, antheridial initial, రెండు antheridial initial నుంచి ఒక్క antheridial initial ఏమో forming the jacket cell, ఇంకొక antheridial initial ఏమవుతుంది, it develops into the androgonial mother cell or the anthrozoids mother cells, which develops into the anthrozoids. So ఇక్కడ anthrozoids ఎట్లా ఉన్నాయి, they have many coils and a single flagellum. So ఇక్కడ చూడండి, this is the antherozoid. It is spirally coiled. ఇదంతా కూడా antherozoid. So anthrozoids etla unnai, it is highly coiled and having a single flagellum. Now the prothalial cell and the jacket cells of both the antheridia, they disintegrate, and all the antherozoids, they become free in the surrounding water. Okay, the antherozoids with the help of the flagellum, they can swim in the water. This is regarding the development of the male gametes from the male gametophyte. Now we'll go for the female gametophyte. Female gametophyte which will be developed from the megaspore. Now, okay, then you go right. So here also you can see the megaspore. It is dividing it into two cells, a smaller cell and the larger cell. A smaller cell occupies the whole beak, and the larger cell, it does not divide further. Only the smaller cell functions as an apical cell, and this apical cell, it divides and cut off and forming a single layered vegetative tissue. Later on, this apical cell only functions as an archegonial initial. Okay, a smaller cell, it occupies, and the larger cell, it does not divide further. Alage untundi, smaller cell ni apical cell annamu. This apical cell will function as an archegonial initial. The archegonial initial divides and produce a small primary cover cells at the top and a central cell at the base. Now, this cover cell, it divides to form the 4 neck canal cells or the initials, and they divide to form a neck region. And the central cell of this, it produces a small primary canal cell and a lower, larger primary ventral cell. Now, within this ventral cell, it will develop and produce a large oosphere or the egg, which is a female gamete. And the primary canal cell, it develops and produces 2 neck canal cells. Now, this ventral canal cell and the neck canal cells of the mature archegonium, they disintegrate and it forms an opening for the entry of the anthrozoid. Endukante venter lo emundi manaki ikkada female gamete undi, egg kabatti, that anthrozoid, they swim and they reach the female gametophyte plant, and they will enter into the venter and fuses with this and forms the zygote. Okay, or that we call it as a fertilization. Now, each megaspore, which is enveloped by a layer of the mucilage. So, in mucilage, anthrozoids are getting attracted towards it. These anthrozoids, they enter into the archegonium and it fertilizes with the egg to produce oosphere.